And myself, Humaira Badruzzaman. I am from J.A.T Arts, Science and Commerce College for Women, Malegaon. My topic is microtechnique. Microtechnique, this technique is used for the formation of permanent slides. So, for many biological studies, the pathological investigation is performed by the help of a technique which is by the help of which the material or slide is prepared by using microtechnique. Microtechnique is the use of number of steps like collection, preparation of material, fixation, dehydration, clearing of material, embedding, cutting of the block, in sections by microtome, dehydration, section staining, and then mounting of section in mount of on a slide. So first of all, we will study about the microtechnique steps one by one. First step is fixative. So fixation is the preservation after death of the shape, structure and chemical constituents of tissue and cells. After death, tissue and cells begin to undergo changes leading to their breakdown and ultimate destruction. Such changes may be due to action of enzymes normally present in the tissues, and so the changes are termed autolytic action or self-destruction. The changes can also be due to external influence such as bacteria which cause decomposition. These bacteria may be constitute the normal flora of the body or pathogens that are present due to disease process. So, to avoid these harmful effects, fixation is necessary. For fixation, the chemicals are used called fixative. The fixative itself has characteristics. Good fixative must kill the cell quickly within minimum of distortion. It must penetrate the tissue and cells rapidly, must insoluble the substance of cells and give good optical differentiation. It must inhibit bacterial decay and autolysis and helpful the tissues to become insensitive to subsequent treatment. It permit the autolysis. It should not add to remove from the tissue constituents and not cause shrinkage or swelling of the tissues. It is not toxic, non-corrosive and non-inflammable. It should be stable and allow for long-term storage of specimens. So these are the characteristics of good fixatives. The fixatives may be chosen as per the need of the investigation. Several fixatives has become established as routine, but for most of the them a specific purpose not suitable for routine use. Other fixatives are used according to their action upon the cell and tissue constituents. So fixatives are also classified as microanatomical, fixatives, cytological fixatives, nuclear fixative, cytoplasmic fixatives. The commonly used fixatives in the laboratory are formaldehyde, mercury chloride, osmium tetroxide, acetic acid, ethyl alcohol, acetone. These are simple fixatives. The compound fixatives are also used by the help of anatomical and cytological fixatives. The anatomical fixatives are microanatomical fixatives present in the form of 10% formal saline, 10% neutral bumped formin, zinc solution, Bowen's fluids. The fixatives are used to immobilize and main objective of it is the maintaining the morphological identity of the tissues. So for fixation of the all parts of the tissues and to change post-mortem changes of the tissues, fixatives are used. The fixatives are used after that which act its color action on the tissues which is not useful for the further process, so its removal is necessary. So for removal of the fixatives, second process is applied which is nothing but the dehydration. Mostly the fixatives are made in the with the help of the water, so removal of the water is necessary. So the next process is nothing but the dehydration, by the help of which, by using some reagent, removal of the water from the tissues takes place and the tissue becomes free of water. This process is called dehydration, in which removal and replacement of water from the tissue takes place by the help of dehydrating agents. The dehydrating agents used in the form of alcohol, acetone, and diethyl dioxide. The During the dehydration, some precautions takes place as dehydration should be done in tightly closed specimen tubes. Specimen tubes should be completely drained before putting higher grade of alcohol. The specimen tube containing the material should be closely tied with glass cork. In case of high humidity in weather, the specimen tube should be kept in desiccators and during the staining, there should be sufficient quantity of stain in the jars so that the slide is completely immersed. So in such a way, dehydration takes place by the help of which removal of the water occurs and the tissue becomes completely dehydrated. After dehydration, the next step used in the microtechnique is nothing but the clearing. This stage of tissue processing can be referred to as dealcoholization, that means removal of alcohol from blocks. This in this To remove the alcohol from the tissue, most of the well-known antimedia have the property of making tissue transparent. Therefore, this stage of tissue processing is referred to as clearing agent, clearing stage, and antimedia is called clearing agent. A good clearing agent must be miscible with alcohol and wax, should remove alcohol quickly and clear rapidly without over hardening of the tissue. So it should be used in proper way for the removal of the water. So the good characteristic of the clearing agent is nothing but the it should be miscible with alcohol. It is easily removed by melted paraffin. It should not produce excessive shrinkage, hardening, or damage of tissues. It should not dissolve in aniline dyes and it should not evaporate quickly in water bath. The clearing agents used in the form of the xylene, toluene, benzene, chloroform, cedar wood oil, and the other materials are also used in the form of the tetrachloride, carbon disulfide, paraffin oil, petrol, etc. So in such a way, the clearing agents are used for the removal of the alcohol. In the case of the dehydration, removal of the water takes place by using grades of alcohol. So to remove the alcohol, the clearing agents are used and the process is known as clearing. Next step is nothing but the impregnation and embedding. These two stages in the processing schedule involve the impregnation of tissue with a medium that will fill all natural cavities, spaces, and interstices of the tissues and solidify to form sufficiently firm that can give considerable support during the cutting of the section without distortion or alteration of the tissues and cellular constituents. The specimen is surrounded with a mass or block of embedding medium. Allow the specimen to be handled and attached to the microtome. The paraffin wax, usually used for embedding, is most popular. Other embedding material include the host of other wax. This embedding material is used in the form of melted form or in the saturated solution. The impregnation is of two types: cold impregnation and hot impregnation. For cold impregnation, the material is used in the form of the wax, which is dissolved in xylene, make saturated solution, keep the tissue in the saturated solution, changes for 3 to 4 times after 10 minutes, and the process is called cold impregnation. During hot impregnation, the melted wax is used, keep the tissue in melted wax, and the temperature of the tissue becomes similar to that of the temperature of the melted wax and able for formation of the block during the block formation. So embedding of the media or embedding of the material is the process which is done after the impregnation, in which tissue or specimen is enclosed in a mass of embedding medium using a mold called L block elements. The tissue blocks are very thin in thickness. They need supporting medium in which the block is embedded. This supporting medium is called embedding medium. Various embedding substances such as paraffin wax, synthetic resins, gelatin are used. The commonly used embedding medium is paraffin wax. Mostly we use the paraffin wax whose temperature is in between 60 to 65 degree centigrade. The embedding media is used which have the quality like which is easily available, uniformity from one batch to another, and show solubility in dehydrating agents, has low viscosity, has good preservation of fine structures and good sectioning quality, able to resistant to heat generated by the sectioning, and stability in electron beam. So these are the characteristics of the embedding medium. The types of embedding media are of two types. One is water soluble, another one is water insoluble. Water soluble embedding medium is present in the form of the gelatin, gums, and polyethylene glycol, while the insoluble water embedding media is present in the form of the methacrylate. In such a way, by using these two media, embedding of the tissues occurs. After embedding, the block making is the another process in which the formation of the block with the help of paraffin takes place. This process is known as casting of block or block making. For the block making, the steps are used as: first, fill the mold or L piece with molten paraffin wax. Then, place the impregnated tissue from in the mold and orient it according to the plane of the section needed. Then they immediately move a warm needle in the molten wax on all sides of the tissue so as to remove air bubbles. Blow the air gently on the mold. The wax forms a thin layer on the surface. Gently immerse the mold in cold water so as to cool the wax rapidly. While immersing, it should be lightly titled to avoid sudden contact with water. When block is solid, remove it from the water and trim it according to need. After removal of the block from the water, its labeling is necessary. The blocks are labeled carrying all the details of the tissue, which is done by inserting the label on one side of the block during casting. The blocks are stored in small paper bags or clean wooden box in cool places until they are sectioned. The next step is section cutting. A section is a thin slice cut from the biological material for studying either the cells themselves or their arrangement. The paraffin T block containing the tissues are cut into sections with the help of microtome. The microtome is also of two types, rocking or rotatory microtome. After that, the next step is trimming the block. The paraffin block is trimmed to give it a correct shape. The paraffin block containing the material is held against light so that the outlines of the material can be seen clearly and then trimmed until the material lies in the center of the perfect rectangle with the major axis of the material. While trimming, care should be taken that the block is not cracked and material is neither exposed nor remain covered with a very thin layer of wax. After the trimming of the block, the mounting of the block is necessary. The trimmed block is now attached to a holder which can be inserted in the jaws of the microtome. The block holder consists of a disc with rough surface attached to a cylindrical rod. The disc is first covered by a thin layer of molten wax and allowed to cool. The paraffin block is now attached to the hard layer of the wax on the disc of the block holder. The block holder with the attached block is allowed to cool and attain room temperature. In such a way, mounting of the block at the to the holder takes place, and after that the block making, labeling, sectioning, trimming and mounting of the block occurs. So in such a way, the microtechnique process is completed. After that, the ribbon formation technique is used.